Psychogenetics. Methods of behavior genetics. Methodological approaches: animal study, twin and family study, measured genetic variants, quasi-experimental design. Animal study. Animal behavior genetics studies are considered more reliable than our studies on humans because animal experiments allow for more variables to be manipulated in laboratory. In animal research, selection experiments has often been employed. Molecular technique. Behavior genetics use wide range of molecular techniques to alter, insert or delete genes. Molecular techniques include knockouts, fluxing, gene knockdown or genome editing using methods like CRISPR. These techniques allow behavior genetics to control in the model organization genomes to evaluate the molecular, physiological or behavior outcomes of genetic changes. Twin and family study. Studying genetics and environmental influences, twin, adaptation and family study. In twin study of twins, both identical or fraternal, really apart and together. Adaptation study. Are adopted children more similar to biological parents or adopted parents? Family study. Study of siblings with different degrees of genetic similarities and varying environments. Measured genetic variants. The Human Genome Project has allowed scientists to directly genotype the sequence of human DNA nucleides. Genetic variants can be tested for association with behavior phenotypes such as mental disorders, cognitive abilities, personality, and so on. Measured genetic variants. Candidate genes, genome-wide association study, SNP heritability and co-heritability. Candidate genes. Candidate genes is selected based on some priority theory about biological mechanism involved in the manifestation of behavior traits or phenotypes. In general, such studies has proven difficulty to broadly replicate, and there has been concern raised that the false positive rate in this type of research is higher. The candidate genes approach. Candidate genes are selected by knowledge of how they influence similar traits in other organisms. There is increasing evidence that some genes can control similar phenotype traits even in distinctly related species. Genome-wide association study. Genome-wide association study (GWAS) look for association with SNVs and genetic factors across the whole genome to correlate with particular traits. SNPs heritability and co-heritability. Researchers has begun to use similarities between classically unrelated people at their measured single nucleotide polymerism SNPs to estimate genetic variations or covariations that is tagged by SNPs. Researchers find that the average genetic relatedness over all SNPs between all individuals in a sample and use Huysman-Elston regression or restricted maximum likelihood to estimate the genetic variation that is tagged by or predicted by the SNPs. The portion of phenotype variation that is accounted for by the genetic variance has been called S&P heritability. Initially, S&P heritability increases to the degree that the phenotype similarities is predicted by genetic similarities at the Maillard S&Ps and is expected to be lower than the true narrow sense heritability to the degree that Maillard S&P fails to tag causal variants. The value of this method is that it is an independent way to estimate heritability that does not require the same assumptions as those in twin and family study, and that it gives insight into the allelic frequency spectrum of causal variants underlying traits variations. Quasi experimental design. Non-equivalent control group design. Non-equivalent control group pre-test post-test design. Interrupted time series design, control series design. So let's just talk about non-equivalent control group design. It's a separate control group. Selection differences, choosing group based on naturally existing categories. This is why study is called non-equivalent. Non-equivalent control group pre-test post-test design. Benefits: a pre-test for confirmation purposes. Interrupted time series design. Measurements of dependent variables is interrupted by the quasi-independent variables. By taking multiple measurements, you can look at the effectiveness of a treatment before and after it is introduced. Controlled series design. Extension of the interrupted time series design with a control group. In general, genetic influences on behavior are passive. Nature of environmental influences, nature of genetic influences. Criticism on behavior genetics. Behavior genetics finding can be challenge social beliefs about the nature of human behavior and abilities. Behavior genetic research on topics such as racial differences, intelligence, violence, and human sexuality.